Good morning, students. Today we are going to continue with the chapter 6. In that, the fourth part we are studying. Before I start with the fourth part, just recalling what was done last class. You have studied about the DNA molecule. You have understood about the double helix structure of DNA. And you have also studied how the DNA will separate, and you know the method, how it goes for the semi-conservative method of DNA replication. It doesn't separate directly. There is a method of separation. So as it separates, the new strand can form. And you know that very well. So imagine if this is the template strand. When I say template, it is like a substrate. So on this strand, I am using the word strand which is having 3' to 5' direction, which is called as template. Why are we calling it template? Imagine if this is the template 3' to 5'. So the synthesis of the new strand is 5' to 3'. Please always remember, direction of synthesis of new strand, direction of the DNA synthesis is 5' to 3'. Okay, I hope it is clear with the previous knowledge. So we are continuing from there. So DNA replication is done. Today we are going to study further of your central dogma of molecular biology. So your DNA produces RNA, and then that gives your proteins. Okay, so today we are studying the conversion, or not conversion, you can say passing the genetic information from DNA to RNA. Okay, passing the genetic information from DNA to RNA. So we are producing RNA. In short, we say DNA that produces mRNA. Produces mRNA is called your transcription. So today we are studying this process, transcription process, in detail. Okay, so replication is done. Today we are studying the transcription. So what is transcription? 1 mark question. If it is asked, you have to write the answer. The process of copying genetic information from one strand of DNA into RNA is termed as transcription. So you know your DNA, A, G, T, C. Okay, so this is your, imagine this is 5' to 3' end. So complementary to this, DNA is double stranded. That's why I'm saying complementary is A is T, G is C over here, T is A, C, complementary G. And this strand will be anti-parallel. So this will be 3' to 5'. Okay, so in this, one of them has the genetic information. Other one does not have the genetic information. Remember, one has genetic information, the other doesn't have the genetic information. To know what is that I'm saying, the one which has the genetic information is called sense strand. Okay, the one which has the genetic information is called sense strand. The one which is having the complementary sequence, example I've shown here, 5' to 3' is sense strand, and complementary to your sense strand we call it as antisense. When I say antisense, it is complementary to sense. Okay. Now in your transcription process, we are selecting antisense strand for your transcription process. Why? I want the genetic information to be expressed over here. So this is your peptide or protein. Okay. So it has to be expressed. How do we express it? We need this genetic information in mRNA. So I can't use this sense strand for transcription, but I can select the the complementary strand, okay, and then use it for production of mRNA, which seems to be similar to the sense strand. Just observe. One thing you have to remember, points to be remembered, wherever T comes, it is replaced to U. I hope you remember what is T and U. In case of DNA, we have told nitrogenous bases, one of them is the thymine. It gets replaced to uracil in case of RNA. So you have to remember, when we are talking about synthesis of RNA, so complementary to antisense strand. So T and complementary is A. A complementary is U. Like that, it keeps producing. Now observe here, it is the template strand we call. Okay, antisense strand is called template strand. Template strand does not code for anything. It does not code for anything. It has the decoded language of sense strand. Okay, it might go above your head for time being. As you study process, you will come to know the idea better. Okay, so now you have to just remember, sense means having the coding sequence. So we call it as coding strand. Antisense, non-coding. Why are we using non-coding? So we can prepare the mRNA which is similar to coding strand. Okay, so mRNA is a coding strand again. Remember, when it produces the coding strand, it is one way of going back to the evolution, preparing RNA, the first genetic material, so that the genetic expression is possible in the present era. So this is what you have to remember. One thing, mRNA will be similar to sense strand. But how do you produce mRNA? By using the antisense strand. Okay, which has the genetic information? Sense. Which is not having the genetic information? Antisense. For transcription, which strand we are using? Antisense. Remember all these terms. So your antisense strand will be from the direction 3' to 5'. So the reading will be in this direction, 3' to 5'. This is antisense strand. We call it as template strand also. So using this template strand, okay, when I say template strand, antisense strand, it produces the transcript. Okay, whatever product we get on transcription, we call it as transcript. Whatever product we get from transcription is called transcript. So remember, transcript is your RNA. Okay, mRNA is a functional one. So you will understand as we go further. So I hope it is clear. So synthesis is 5' to 3'. Whatever mRNA you are getting, the transcript is 5' to 3' direction. So synthesis, again, synthesis is always 5' to 3'. This should be registered in your head, in your mind. So we'll go into the topic of transcription. Certain things which you have to remember about transcription. First, the principle of complementarity governs the process of transcription. Of course, you understood how this antisense strand produces the mRNA based on the complementarity. Okay. So instead of adenosine that pairs with thymine, in case of RNA, it is pairing with uracil. Okay, so we have A that pairs with T in case of DNA. But in case of RNA, what it pairs with? A pairs with U. Okay, remember. So that's what we are saying. Adenosine forms base pair with uracil instead of thymine. Okay. And in your transcription, here you observed, I said only antisense strand is used, not the sense strand. the question comes, why can't it be? Okay, so that's what we have told here, only a segment of DNA and only one of the strand. So when I say segment of DNA, you know the length of the DNA is very long, right? Which you have studied already. So it is almost 2 meters long. One chromosome has the length of the DNA almost 2 meters long. So you can say this part is one gene, this part is another gene. So I'm talking about one gene that is starting to express. What do you mean by expressing? It undergoes transcription, it undergoes translation. Finally, the important protein is formed, and this protein is used for metabolism or for any activity. It may be like hair color, hair growth, hair texture, eye color. So it is based on the protein that is formed. So this is called as gene expression. So that's what we are saying. Only a segment of DNA. What do you mean by segment of DNA? That codes for a character. So it is your gene. Okay, this you have to remember. That's what it's saying. Transcription is only a segment of DNA. So at a time, at different places, so at a time, imagine this is the, sorry. So imagine if this is the DNA, I just roughly drew. This portion undergoes transcription, even this portion undergoes transcription. So like that, in the long DNA strand, at different locations, transcription can occur. So that's what you have to remember. A small segment and on one strand. Which strand am I talking about? Particularly in antisense strand, transcription occurs. This is the term which we use for the past tense. Transcribed, transcription. Okay? So this is your process. This is your past tense, transcribed. So whatever product you get is transcript, remember. Okay, if you just cut ION, it is your transcript. That is the product. That is your RNA. Okay, the question if it is asked why both the strands are not copied during transcription. First you should know, if it does so, RNA molecule will have different sequences. So when I say RNA, I'm talking about the mRNA, okay? So mRNA, which was supposed to express or produce proteins, will have different sequence, which will again produce different different protein itself. So the sequence of amino acids in protein also will be different. You'll understand this better when we study translation. Okay, so whatever your sequence is there in the antisense strand is important for your genetic information to be expressed. In case if you're taking the sense strand, the sequence of amino acid will change, the sequence of the sense strand is different, therefore even RNA will be different, and whatever protein you get is different. This is why they are saying they would complicate the genetic information transfer machinery. So whatever we needed to express the character, okay, that is your genetic information, that is making it difficult to happen. Okay, if both happens, if both the template strands are transcribed. What is the second reason it does not allow that? See, as soon as one strand is formed, 5 prime to 3 prime, the DNA other sense strand also produces 5 prime to 3 prime. Okay, and it will be complementary to each other, because from DNA also there were two strands which were complementary to each other. When they produce the RNA, definitely these two will be, the whatever RNA transcripts we get will be complementary. Hence, they will form a double strand. Immediately those two RNAs will form double strand, and it will be of no use. It will not produce any protein. It will not produce any protein. That's why, to avoid this problem, we are saying it should be only on one strand, and compulsorily on antisense strand it should transcribe. So I hope the idea is clear. Now you should know about what transcription unit is. Now you know where transcription occurs. In the nucleus, of course, you know your cell, you have your nucleus, you have your DNA, and from DNA you are getting your RNA. But you don't know which is the location of that transcription. So we call for each gene, we call it as transcription unit. So that gene has 3 particular regions. So we are saying transcription unit in DNA it is. In DNA the unit is present. So that unit has 3 regions. First is promoter, second is structural gene, third is terminator. So you will come to know the term itself gives you some idea. Promoter, it promotes the transcription. It gives a startup. Structural gene, the gene which is supposed to be expressed. It has the sequence coding for a protein. Okay. Then there is your terminator gene. Terminator means termination, end. Okay, it is an end. So this is what you have to remember. 3 regions are there, which is your transcription unit. If it is asked for 2 marks, mention the 3 regions of transcription unit. Please remember to mention these 3 points. If it is asked for 3 marks, that means you have to highlight the structure. So you have your DNA here. This whole structure is your DNA. Template strand, coding strand. This is from your textbook. This is actually a picture. It's a little modified. Just observe here. There is a promoter region. There is a terminator region. There is your structural gene. So you got your 3 genes. But first you should know what is this DNA consisting of. As I said earlier, template strand, coding strand. When I say template, we are using that strand. Template means we are going to use that strand. So that template acts like a substrate. So this is the one which we are using is your antisense. Okay, this is your antisense strand. Now, what about the coding strand? Yes, you know very well. Now coding is your opposite to antisense is sense strand. So you got some idea? Sense, antisense. So you don't get confused. We are selecting the antisense strand which is in the direction 3' to 5'. The arrow is shown properly. Just don't get confused. Arrow is proper here. We are saying how the bonding takes place. It is 5' to 3', 5' to 3'. Okay. Bonding is what I'm talking about is phosphodiester bonding for the DNA nucleic acid, the polymer to form. Now, this is your template strand which is 3' to 5'. This is your coding strand, the sense strand, which is 5' to 3' here. So we are talking about mainly the template strand. Okay, and the complementary strand is your coding strand. So we are taking template above, coding strand below. And we are saying transcription is in this direction. That's why we have shown a black arrow here. Transcription is in this direction. So how does the transcription begins? It begins at the promoter region, which is at the 5' end of coding region. Sorry, coding strand, not region. Coding strand. Okay. Region comes somewhere else. That I'll explain you later on. Please remember the word is strand. This is your coding strand. And then in case of template strand, it is beginning from the 3' end of the template strand or antisense strand. So you can select any one which is easy for you. So this is one example I've given. So this is your template strand and complementary to it is your coding strand. This is completely DNA. There is no RNA shown. Okay, there is no RNA here. I hope you've got the idea. So this is your antisense strand, the template strand, which we are taking into consideration for transcription. Opposite to it is the coding strand. So there is initially the promoter, then there is your structural gene, and then there is your terminator. So you got the idea? This is from your textbook. So important question. This is what you have to practice. Is that clear? So transcription unit is clear. 3-mark question. Draw the schematic structure of a transcription unit. So you have to only draw. Okay. Majorly asked is your schematic structure. Never asked for explanation. Just in case if they separately ask what is promoter, what is structural genes, what is terminator, you can give in your own words, but it is better you know some knowledge. So some points to remember. When I say promoter, it provides the binding site for RNA polymerase. Where is that site, binding site? On the DNA. So you remember this place. This place. It is on DNA. Okay. So there is one enzyme called RNA polymerase. Now remember, RNA polymerase. Polymer forming enzyme. Polymerase. What we are producing? RNA. That's why the term for the enzyme is RNA polymerase for your transcription. So you recall back to DNA replication. There it was DNA polymerase. Here it is, since we are producing mRNA, so now we are talking about RNA polymerase. So now you understood the promoter. It provides the binding site on DNA. Okay. Binding site for whom? It is for the RNA polymerase. So where is that promoter region present on that sequence? It is at the start site. It is a start site where I say in case of your coding strand, which is a sense strand, it is 5'. If it was your template strand, then it is 3'. It is left to you how you want to answer. Which is easy for you, you can follow. You can follow this itself, no problem. But you should know the concept, from where it begins and which strand we are using for transcription. Then you should know about the structural genes. So which is the part we are reading? We are reading the non-coding strand or the antisense strand in the direction 3' to 5'. This is the one, 3' to 5'. Direction of reading. This is the structural gene. So it is your non-coding. Opposite is coding here. Okay. Template strand is non-coding or your antisense strand. And you know very well, whatever transcript you get after reading, it is your 5' to 3' direction. Okay. Let me show back over here. So you remember this one, 3' to 5' direction, and complementary to the antisense strand. And how it is pairing? A pairs with U. Instead of A pairing with the T, A pairs with U. Okay. Now